Okay, so let's get started. So this is regarding the yeast cloning vectors. So in my last class, I had covered some of the plasmid vectors, then modification of the plasmid vectors to accommodate larger insert sizes. And then again, I had told you that there are something called as back vectors. And I also covered a part of a yeast cloning vector, which is YAC vectors, and how some modifications are being made in YAC vectors to make the insert in them more stable. So let's say in continuation with that, I'm continuing with the yeast cloning and expression vectors. So with respect to bacteria, if you remember that there are two ways by which you can transfer a particular gene of interest into bacteria, which is either with the help of a plasmid or by viruses, bacteriophages. And in case of yeast, do we have any plasmids which exist in yeast? So yes, some of the early these yeast vectors, they were based on a natural plasmid, which is 2 micrometer plasmid is the name that was given to that. So it is a naturally occurring plasmid. In general, its copy number is around 20 to 80 in a yeast cell. And also the features of this plasmid, I'll just introduce you to. So like any other vector, it has these replication genes. It has this flip region. And does it have any importance with respect to cloning? No, it doesn't have with respect to any cloning. It is just that I'm telling you the features of 2 micrometer plasmid. So replication genes, there is a flip region. I'll just come back to this. And then there is some D protein. There is origin of replication. Now, the flip region, it on the either side, what we have is the inverted repeats, and it is the recombinant, which are means completely these complementary with each other. They are two perfect inverted repeats. So when the recombination takes place between them, then the region between those, it gets flipped. So that's why in some 2 micrometer plasmid you will find the flip region in a particular direction, while in others it will be present in the opposite direction. So this is the structure, and this was the basis of the rest of the yeast cloning vectors that were developed subsequent to it. So when we talk about the yeast vectors, they are divided into three, rather four, or rather five different categories. So these three are the ones which are, let's say, given in TA Brown. Then there is something also called as yeast centromere plasmids that I'll be covering. And then the last category can also be the YAC vectors, which are the yeast linear vectors, in which the ends are free, they are not circular in nature. So coming to these three plasmids one by one. Okay, so yeast episomal plasmids. So what are the features of this plasmid? As you can see, it has origin of replication derived from the 2 micrometer plasmid. Then it is a shuttle vector. If it is a shuttle vector, it means that it can also grow in another host. Which host it will grow in? For which it has the origin of replication. In this case, it has a plasmid origin of replication. So it means that this particular plasmid can stay in two different hosts. One is yeast, one is bacteria. So if we want to, let's say, manipulate this particular plasmid and introduce our gene of interest, then obviously we first make the plasmid and introduce it into bacteria. And for to check for the recombinants, how do we do the selection? With the help of certain antibiotic markers, and in this case it's ampicillin or tetracycline. Then do we have any selection marker for yeast? Yes, URA3 is there. So in my last class, I had introduced you to the fact that if you want to do selection in case of yeast, then generally the auxotrophic strains of yeast are used, and the plasmids have the genes which are helping in the synthesis of that particular amino acid or whatever product is there. So in this case, the plasmid has URA3 gene, which is involved in pyrimidine biosynthesis. So the yeast strains which take up this plasmid, they will not grow in the absence of pyrimidine, but if they have not taken up the plasmid, but otherwise they will grow. So yeast episomal plasmids, their copy range is around 20, and they are quite unstable in nature. Then we have yeast integrative plasmid. So let's see, what does this plasmid have. You can notice that there is one plasmid origin of replication. There are the two antibiotics which help it being maintained in bacteria for selection purpose. And then we have this URA3 gene. But you will notice that even though it's a yeast plasmid, it doesn't have any yeast origin of replication. So it means that this particular entity will only survive in yeast if it gets integrated into the genome. So how does it get integrated into the genome? So they use a yeast host strain which has URA3 gene, and this URA3 gene then so this URA3 gene is also present in the plasmid. So because they both are homologous, the URA3 genes, so there is a chance event of recombination between those, and then this plasmid can get integrated into the yeast genome. So that's why they are called as yeast integrative plasmids. Then coming to the yeast replicative plasmids. So here what do we have? We have a plasmid origin of replication, ampicillin and tetracycline for selection in bacteria. Then we have tryptophan for selection in yeast. And what else do we have? That rather than the origin of replication of the plasmid that exists in the yeast, we have autonomous replicating sequence which is required for replication of yeast chromosome. And if you remember, I had introduced ARS1 also as part of the YAC introduction, YAC plasmids, YAC vectors, sorry. So this is the structure of yeast replicative plasmids. They have comparatively much higher copy number as compared to episomal or integrative, but these are also a little bit unstable in nature. Then let me compare these three, and then I'll just come to just briefly introduce you to the centromere plasmids. So when we talk with respect to the transformation frequency, the episomal plasmids have the best transformation efficiency as compared to replicative or integrative one. Why the transformation efficiency of YIPs is extremely low? Because of a basic fact that they are totally dependent on integration into the yeast chromosome. Okay. Then comes the copy number. I told you that YEPs have around 20 to 50, RPs, that is the replicative ones, have up to 100 copy number. But these integrative plasmid, you can understand, because they are integrating into the genome, their copy number is just 1. Okay. So their copy number is very, very low. But in those cases where you want to maintain a particular stretch of DNA inside a vector to be, maintained maybe in the form of a library or something. So in those cases, YIPs are very, very good, because obviously because it's an integration event, they are extremely, extremely stable in nature. So once it's integrated into the chromosome, its excision from the chromosome is a very, very rare event. So that's why they are highly stable in nature. Okay, so that's what is written here also, that YIPs are very, very stable, and RPs, the replicative plasmids, are very unstable, and YEPs also have similar problem, but now people are manipulating EPs and RPs and trying to make them much more stable, because now we have a better understanding of the various genes that are involved in providing stability and so on. So improvements are being made in these vectors. Now coming to the centromere plasmid. So centromere plasmid also, let's see what does it have. It has an origin of replication for plasmid. It has some bacterial antibiotic marker. It has some yeast antibiotic marker. And obviously this your favorite gene is just the YFG full form. Means you clone your gene of interest here, which is obviously the case in the previous plasmids as well. It has the autonomous replicating sequence, but apart from that, it has a centromere sequence. So it means that this plasmid is comparatively much more stable, and the presence of centromere will ensure that its distribution in the daughter cells is comparatively much even as compared to if centromere was not there. So these are the yeast centromere plasmids, and with respect to the linear plasmids, what they have done is they have added telomere sequences. That these these vectors can also be maintained as linear chromosomes by the presence of not only the replicating sequence for the chromosome, but also with the help of centromere, and which will ensure its equal distribution, and telomere on the either ends, which are highly GC-rich sequences, and protect the chromosome from degradation. So that's all for the introduction to the yeast plasmids. Now coming to that, like in bacteria, I introduced you to the strain which is commonly used for cloning purposes, and in fact, there was a very detailed class on the various strains and what changes have been made in bacterial strains to make them highly, very good host strains for expression purposes. It was one of my early classes. So similarly for these yeast, we have various strains which are used. I'll come back to the various vectors, various promoters that are used for expression of a particular gene in yeast. But let me first introduce you to the host strains. So S. cerevisiae, जैसे like in bacteria you had E. coli, so very easy to maintain and very fast growing. So similarly for S. cerevisiae, it is comparatively a very easy strain to work with, and second point is that like E. coli, even its genome is completely sequenced. Now we know the function of many genes which are present in S. cerevisiae. So overall, we pretty much know the function of almost all genes which are present in this. So even the manipulation of host strains for betterment of the expression of your gene of interest is easy here. Now, is it the best strain that is being used in the companies or in labs for protein production purposes? No. It is used in those cases definitely where you want to overexpress a eukaryotic protein, and you find that E. coli was not giving much of the activity of the protein for various reasons. But in case of S. cerevisiae, because it's a eukaryotic system, some of the modifications which were not happening in E. coli, they can happen in S. cerevisiae. But at the same time, with respect to glycosylation, it was realized that in case of S. cerevisiae, it rather is hyperglycosylating. So I have one picture here. So this is, let's say, some protein which is expressed in human. And this is the glycosylation pattern here. But in S. cerevisiae, when I overexpress the same protein, I see huge amount of hyperglycosylation here, right? So what will happen if I express this protein in S. cerevisiae, and then I give, let's say, that protein for therapeutic purposes in to some patient, then because it is not similar to the human protein, it can be taken as foreign, as a foreign protein, and then it may lead to the induction of the immune system. and there is a possibility that there can be this immune attack on these proteins, and which is definitely not good, and that's not what you are seeking. So in those cases, S. cerevisiae is not preferred, and also it was realized that because it does not have a good system for secreting proteins into the growth medium, then the way to harvest the final protein, your recombinant protein, is comparatively difficult as opposed to when you just have to harvest the protein from the media in which the yeast was growing. Okay. So in that context, Pichia pastores has gained a lot of attention of late, and is being used already for various for production of various recombinant proteins. And the reason is simple, that as compared to S. cerevisiae, Pichia pastores makes the proteins which have very similar glycosylation patterns like the human proteins. Okay. And second thing is that it has a very good secretory system, and you can harvest the proteins from the growth media. So these are the two major systems. In our department also, I see that many people have now moved to Pichia pastores. I know Professor Saroj Mishra's lab. Now I think even Professor Atul Narang's lab, they are working on this, and maybe so means I'll just come to that, which are the other strains of yeast that are being used. So I also don't know that so many names have come up with respect to yeast, because I told you it's a dynamic system. People are looking for ideal host strains which have all the good properties of functioning as a host strain. And which are the good properties? I've discussed those in context of bacterial host strain introduction as well. So these are some of the new new host strains of yeast that people are working on. And I know that one of the faculty, I am not remembering, could be Professor Ashish Mishra. I think he is trying out this particular strain for production of recombinant proteins. Now coming to that, when we are using yeast as the system for cloning purposes, mainly of eukaryotic genes, then obviously in this system, we want to express the protein and get the final protein out. To express any gene, what we require is a very good promoter. So with respect to the plasmid vectors, I'm yet to tell you that which are the inducible promoters or continuous promoters that are generally used, or in context of mammalian vectors also. I'll just come to that. But in case of yeast strains, I thought, because the class is completely on yeast strains, so let me tell you about these promoters. So these are the very generally used promoters which are cloned upstream of the gene of interest. Okay. So these are generally present in the various yeast vectors which are commercially available, and you have to clone your gene of interest downstream of it. So these are some of these, they are inducible promoters like galactose. So GAL10 promoter, it is only induced in response to galactose and gives a very high expression. AOX promoter is maybe the most used promoter in context of yeast, and it's used in those yeast strains which use methane as a carbon source. So when you give methanol, this gives a very huge induction in the protein. I mean, that the promoter gets induced, and then you have a very high expression of your gene. Then you have glucoamylase promoter, which gets induced in response to starch, but in response to xylose, there is no transcription. So means if you want to switch on and off, let's say, the expression of your gene, this is one of the way by which you can do that. Then cellobiohydrolase promoter gets induced in response to cellulose. So this is just an introduction to the various inducible promoters which are used in the yeast cloning system. And in case of bacteria, I've told you the IPTG inducible system, but I'll introduce you to others when I'll be coming to the expression vectors. So that's all the introduction to yeast. So I've told you about the yeast host strains that can be used. I've told you that which are the various vectors which are there, and also I've introduced you to the various promoters that are used when you want to clone your gene of interest in yeast. And why you want to prefer yeast over bacteria, we have discussed it many times. Mainly because if you want to go for eukaryotic protein expression, then you will prefer a yeast cloning vector as compared to bacterial cloning vector. So now I will come to the cloning vectors for plants. Okay, so in case of plants, do we have a naturally occurring plasmid? We don't have any like in bacteria or yeast. So, but is there some way that a plasmid can be transferred to plants? Yes, there are ways. So let's come to the three strategies that are generally used for the cloning vectors for plants. Why you want to clone in plants? Various purposes. You want to make a transgenic plant, you want to make a drought resistant plant, you want to make a plant more beautiful by transferring a gene, you want to make a plant more nutritious in nature, or even, you know, if you want to even plants are now being used as I don't know whether you've already heard, but there's something called as plant vaccines, in which the idea is to overexpress a protein for consuming which it will lead to induction of immunity in the humans. So plant vaccines is also something that is coming up for basic understanding purposes. You want to know the function of any gene, then you have to either overexpress a gene or you want to knock it down. So for those purposes also you have to make transgenic plants. And in all these scenarios that I'm telling you, what you finally want to do is you want to introduce your gene of interest into the plants. Now the whole thing how you can introduce is can be done through cloning vectors, and the classes on that, and which are the three ways by which gene of interest can be transferred to plants. One is through agrobacterium. I'll come to this agrobacterium based plant transformation. Then there is also a possibility of directly transferring a gene using DNA fragments into the plants. And finally, like in case of bacteria, if you remember, the transfer could be either through viruses or through the plasmids. So similarly, in case of plants also, we do have certain plant viruses. The knowledge of which became means we came to know that they exist when we came to know that okay, there are certain plant diseases. Then we came to know why these diseases are there. Then it turned out that it has certain viruses. So it means that there is some way that these plant viruses are transferring their genome into the plants. So yes, even these can be used as the plant cloning vectors. Coming to the first strategy, which says vectors based on naturally occurring plasmids of agrobacterium. So it means that in the plants, the plasmid can be transferred from agrobacterium, from a bacteria called as agrobacterium. So its full form is Agrobacterium tumefaciens. So what it does is that, let's say this is a healthy plant and there is some wound in the plant. So from the wound, certain chemicals are released. It is seen that agrobacteria, it gets attracted by those chemicals. It goes there and it has some way, I'll tell you how it does, of transferring its plasmid, its native plasmid called as TI plasmid into the plant cells. And once that plasmid is inside the plant cells, it somehow promotes the division of the plant cells, and as a result it leads to the presence of a crown gall. Means the cells have so much divided in this region, so fast division in this region, that it leads to presence of almost a tumor type of thing here, and this is called as crown gall. So that's why this disease was called as crown gall disease. That is, agrobacterium can lead to induction of a crown gall disease in certain plants. So, what was the plasmid that I'm talking about that is transferred from agrobacterium to these plants? That plasmid is TI plasmid. Why is it called TI plasmid? Because it leads to induction of tumor. So, it's called as tumor inducing plasmid. What is the size of this plasmid? Around 200 kb. What are the various regions which are present in this plasmid? Let's see. So this plasmid has its origin of replication. Okay. Then to be maintained in bacteria, which is agrobacterium in this case. Okay. Then it has this tDNA region. Why is it called tDNA region? Because it stands for transfer DNA region. Why transfer DNA region? Because it is the region which is present between the left and the right border of the tDNA that gets transferred to the plant cells. Okay, so this is tDNA or transfer DNA region. What are the components of this tDNA region? A left and a right border, which is essential for transferring of the transfer DNA to the plant cell. And I think majorly it's the left border which is responsible, but yes, both of these are important. Then apart from that, there are three different set of genes which are important. One codes for a plant hormone called auxin. One codes for a plant hormone called cytokinin, and one codes for opines, various opines. So this region which codes for auxin, so auxin is a hormone that leads to induction of roots. Okay. Cytokines is a hormone that leads to induction of shoots. So if any plant cell gets very high amount of both auxin and cytokinin, it's the ratio that is important. So if let's say a plant cell gets very high amount of auxin, then what will happen? It will lead to induction of root. If a plant cell gets very high amount of cytokinin and low amount of auxin, it will lead to induction of shoots. But if a plant cell gets very high amount of both of these, it will lead to induction of tumor, means excessive division of the plant cells and no specific induction of shoot or root, but yes, it will lead to tumor. So these two are there. Then opines. Opines is something that is being used by the bacteria to thrive in that region. So this region also means in a way this region is being sent by the bacteria to the plant cells so that it also induces opines. How does it ensure that very huge amount of opine is being produced, which the bacteria can utilize for its own growth, is by a lot of cell division in plant cells. So more plant cells, more with this tDNA region, more opines are being made. So these three are the regions. Then to catabolize the opines, it does also have the opine catabolism region in this plasmid. So yes, so these are the components. Now coming to something which is written here, which is virulence region. So this is called the vir region, and this region encompasses a whole battery of vir genes or virulence genes from A to G, and the function of each of these is given here. So you can please see it. But I can tell you in nutshell that these genes are important for transfer of this tDNA region to the plant cell. If the virulence region or the products from the virulence regions are not there, then TI plasmid cannot be, tDNA region cannot be transferred to plant cells, because this is the region that is involved in making. We can just briefly go through it. One of the product makes the pilus-like structure. Then one leads to transfer of tDNA. One leads to products which lead to excision of the tDNA fragment, and then one product leads to protection of that tDNA fragment which is transferred from nuclease attack, and so on. So you can please go through this slide. So this is the whole structure of TI plasmid, and now we understand that why when the agrobacterium infects the plant cells, it leads to crown gall disease. Okay. So now coming to can we use this TI plasmid as such for cloning purposes? Yes, why not if we can clone something within this region, right? So then if I have my gene of interest in this region, then it can be easily transferred to the plant cell. But the problem is that TI plasmid size is 200 kb. So if the size is 200 kb, it will be very, very difficult to to manipulate this plasmid because it's quite big in size. We always prefer plasmids which are small in size and thus comparatively easy to manipulate. So one problem is that, and second is because the size is very big, how will you find unique restriction sites for transferring of your gene of interest into the TI plasmid? So what you do is that you can use any of these two strategies, the binary vector strategy or the co-integration strategy. So so these two strategies, they make it easy for cloning our gene of interest into the TI plasmid. So let's come to the binary vector strategy. So here what they have done is, in a way, they have divided their plasmid into two sections, okay, the TI plasmid. So one plasmid is only 20 kb and has tDNA region inside it. And one plasmid has rest of the genes because unless, so even TI plasmid also has host specificity region, which somewhere tells you that if this region is there, it means that the agrobacterium cannot infect all the different types of plant cells, but rather it has certain specificity. So this is for your information. And obviously, if virulence region is not there, this region cannot be transferred. So what they have done is that they have, in this plasmid, you clone your gene of interest. They have made a unique restriction site because the tDNA size is only, I think, around 50-60 kb. No, sorry, it's 15 to 13 kb in size, and there are around 8 genes. So in this tDNA region, there is because it's very small in size, you do have option for a unique restriction site. So you can clone your gene of interest here. Once you have your gene of interest here, then what you do is that you transform the agrobacterium with both of these plasmids. So what will happen? Only thing is that now agrobacterium has its plasmid divided into two regions. So this plasmid will lead to the where products being made. These products will ensure that the cut is made on the either side of tDNA and this region is transferred to the plant cell. So once inside the plant cell, I think I missed that. So once inside the plant cell, tDNA, it gets integrated into the plant chromosome DNA. So where does it get integrated into the plant chromosomes? It's quite random in nature. Do we understand that what leads to its integration into the plant chromosomes? Is it homology based? No, it's not homology based. It's very, very random in nature, and people are still trying to understand that what guides that at what region the tDNA will get incorporated. What's happening that if we clone our gene of interest here, then there is a very good chance that it can be integrated into the plant chromosomes. So this is the binary vector strategy. I hope it is clear. And then coming to the co-integration strategy, again a smart strategy. What they have done is that they have taken a very small vector. It's a plasmid vector, like PBR types of vector that I told you before. where we have all those genes. It will have a ORI to be maintained in bacteria. It will have these antibiotic resistance genes and so on. It will have some unique restriction enzyme sites for your gene to be cloned. And apart from that, what they have done, that in the PBR plasmid, they have added a very, very small fragment of tDNA. Okay, so manipulation of this plasmid is very easy. Like any plasmid that you have in the lab, you are introducing your gene of interest, you will just do it like that. Then in the bigger plasmid, the TI plasmid, what they have done is that you incubate these two together. So what will happen, that because there is a tDNA fragment which is homologous to this tDNA fragment, there will be a recombination that will take place. If the recombination takes place, then this whole plasmid will get integrated within the tDNA region. So it means that if the problem I was facing for manipulation of this big plasmid has been solved by this co-integration strategy, and I could transfer my gene of interest into the plant vector, into this TI vector. And this TI vector I will introduce into the agrobacterium. Once inside the agrobacterium, I will incubate my agrobacterium with the plants, and then it will be able to transfer the tDNA into the plant cell. So what other changes they have made in the TI vectors? So what they have done is that this is, let's say, the more very used binary TI vector. So as compared to co-integration strategy, I think binary vector strategy is easier because it doesn't rely on the recombination event. And one of the binary vector is shown here. So what it does is they have disarmed the TI vector. So what they have done is that they have removed all the cancer genes. If you remember, it had the genes coding for auxin as well as cytokinin. So they have removed those genes from the TI vector, and also they have retained the ends of the tDNA. So if the ends of the tDNA are maintained, then where genes don't care what is there inside, they will make a cut on the either side and then transfer the tDNA region into the plant cell. So these vectors, from which the cancer genes have been removed, they are called as disarmed TI vectors. So how do you make a transgenic plant is the basic question. Let's say that I make my agrobacterium strain and introduce my gene of interest in the TI plasmid there. Okay. So what I will do is that, let's say I want to make a transgenic or transgenic plant. I make a cut here and I incubate my agrobacterium here. So agrobacterium will transfer the tDNA here, and let's say I have not yet disarmed my TI plasmid. So it will lead to the transfer of the clone gene here, and which will be only present in the plant cells which are as part of the crown gall or the tumor present, right? But this is not what I'm looking for. My aim is to make a whole transgenic plant in which every cell of the plant is having my gene of interest. Or it could be that in some case that I want my gene of interest to be expressed only in leaf. So how will I ensure that my gene of interest only is expressed in some particular plant of, gene of in any particular part of the plant is, what I can do is I can use promoters which are specifically expressed in a particular plant part. So there are some root specific promoters, there are some leaf specific promoters, some are shoot, some are let's say stamen specific promoter. So all those things, that option I have. But still, what one thing that I definitely want is that my gene of interest should go in maximum uh, in the area that I'm interested in, and not limited to the just the plant cell which the agrobacterium infects. when I'm growing the solid plant. So to avoid that problem, what is currently used is that they take plant cells. Let's say they make a plant callus, or they macerate any part of the plant, and they make a cell suspension. So it means my plant cells are now present as a single cell in a suspension media. I incubate these plant cells with the agrobacterium, the recombinant agrobacterium, in the sense that the plasmid in it, the TI plasmid, is recombinant with my gene of interest. So this agrobacterium will then infect the plant cell in presence of those chemicals that promotes into the plant cell. So once it's inside the plant, is that I place these plant cells on a solid plate, solid media, and I give them high amount of cytokinin from outside. So it means that this media is enriched in cytokinin. Once it's enriched in cytokinin, what will happen? That it will induce the expression of shoots. So what will happen? That the shoots will be formed, and at the same time, after some time, what I add in the media are the root inducing media, which is the media which is high in auxin. So what will happen? Cell which had taken up the plasmid, and in presence of maybe some selection which is present in this plate, that only the plant cells which have taken up the bacteria, they survive. Those plant cells have been induced to form a callus by growing them. Sorry, first they are grown in presence of both high auxin and high cytokinin. So what will happen? Because both the levels are high, it will lead to callus induction, more like tumor induction. Once we have that, तो एक cell से इतने सारे cells मिल गए. Now I want my plant to be regrown. I want to make my transgenic plant. So what I do is that I first induce the shoots by growing them now in the media which is high in cytokinin. So this callus will give rise to shoots. And after some time, I grow them in presence of high amount of auxin. So all these notes are present in the in some link or the note section. So then I give them the auxins, and then these will give rise to roots. And this small plant, I then transfer it to soil for it to become a full-grown plant. So it means from one plant cell, I could regrow my whole plant, which was not possible in this scenario, because here my gene of interest would have been transferred to only select cells which I don't want. Only thing is that if in my cloning vector, that is TI vector or whatever binary vector that I'm making, I use the promoter, which is constitutive in nature, high expression, then my gene of interest will be present in all the parts of the plant. If I use a promoter which is leaf specific, then even though my gene has gone in all parts of the plant, in all cells of the plant, but still my gene of interest will only be expressed in the leaf. And if I use my promoter which is present, which is expressed only in the petals region, then it will be present only in petals, even though the gene is present in rest of the plant cells as well. Okay. So this is one way, by use of agrobacterium tumefaciens, how you can use TI plasmid for as a cloning vector for transfer of genes into the plant cells. So is agrobacterium tumefaciens the only strain that is used? Yes, it's one of the major strain that is used for transfer of the gene of interest to the plant cells. But one problem with the agrobacterium tumefaciens is, as well as the other strain that I'll just come to, agrobacterium rhizogenes, is that it only infects the dicots. If you remember, in the TI plasmid, I had introduced you to host specificity region. Because of that region, it was seen that this particular plasmid or agrobacterium strain it infects the dicot plants very, very efficiently, but for monocots it was not a good vector to be used. So it was realized that while this can easily infect the dicots, the monocots were not very easily transformed by these agrobacterium strains. So if you have to make a transgenic wheat, barley, rice and maize, all of which are monocots, then this was not the preferred system. At least I remember during the time when I was doing my PhD during late 1990s or 2000. So that time the method that was used was particle bombardment. I tell you that which are the various methods which can be used for transfer of genes to the plant cells in my next class. But yes, this was a method that was generally used that time. But now I know that in many labs which are making these wheat, rice, maize, etc., they don't use these particle bombardment, but now they are also using agrobacterium tumefacients because now some changes have been made likely in the host specificity region, by which A tumefacients can also infect these monocot plants. So that time it was considered that, wow, if you're working on dicots, it's not a problem. Agrobacterium will very beautifully transfer your gene of interest there. And people had to resort to particle bombardment, which was difficult with respect to the success rate also. But now many labs have started using the different these altered strains as well as the vectors, which are pretty good for monocots as well. I missed this slide which is on agrobacterium rhizogenes. जैसे agrobacterium tumefacients था ना, उसी तरह से there is agrobacterium rhizogenes, and if you remember, the tumefacients would lead to crown gall disease. The RI plasmid, which is present in agrobacterium rhizogenes, it gives rise to hairy root disease. So means more hairy roots are formed. So if you want to, just infect your gene of interest with agrobacterium rhizogenes, which has a recombinant RI plasmid, then your gene of interest will be very, very highly expressed in all the root cells of the plant. And also because these roots are now much more in number as compared to the parent plant because of the hairy root disease that it induces. So if you want to harvest your protein from the roots, you can do so. And also so many people they also resorted to one of the method in which they were growing the plants which were infected with agrobacterium rhizogenes in the hydroponic media. Means the plasmids are now in a liquid media and they grow their plants, they have their protein of interest in such a way that it is secreted out of the roots. So what happens that when you're growing your plants, transgenic plants in hydroponic media, you can just harvest the media in which your protein of interest is being expressed. So this is also one of the way that many people are working on agrobacterium rhizogenes. But in general, molecular biology labs or plant molecular biology labs, I don't see agrobacterium rhizogenes as a generally used one. It's definitely A tumefacients that mostly people are using. So the rest of the two strategies of direct gene transfer or the use of plant viruses, I will continue in my next class, but I'm open for questions for the class today. Are you guys there?